This is Module 9, End-to-End Procedural Responsibilities in the Lab, Pre-analytical, Analytical, and Post-analytical phases. Once again, this session is approved for crew credits by the AAFP. At the end of this module, you'll be able to understand the testing life cycle, explain the pre-analytical, analytical, and post-analytical phases of laboratory testing and their impact on test accuracy and patient care, establish workflow oversight, coordinate workflows across all procedural phases to identify and eliminate inefficiencies, bottlenecks, or potential risks, maintain documentation, ensure accurate and complete documentation of all laboratory procedures, tests, and corrective actions for regulatory compliance and quality assurance. Introduction to the laboratory testing phases. The overall workflow includes the pre-analytical phase. This is where specimens are collected, transported, and prepared for testing. The analytical phase. This is testing and measurement activities. And the post-analytical phase. Reporting and follow-up actions. So your pre-analytical phase, pretty straightforward. It's whatever you're doing before the sample is being tested in the laboratory. And then your analytical phase is all the testing procedures that go through in the actual testing of the specimen. And then post-analytical phase is the reporting and other QC documentation. Why the phases matter. Each phase impacts the overall accuracy and reliability of patient test results. Errors in any phase can compromise the diagnostic process. It's a collaborative effort. Effective laboratory operations require coordination among healthcare providers, lab personnel, and support staff. Focus on quality. A systematic approach ensures compliance with standards and minimizes errors. Now let's dive into the pre-analytical phase. The definition of a pre-analytical phase includes all processes from test ordering to the point the specimen is ready for analysis. This includes healthcare providers. They ensure proper test selection, accurate patient information, and correct sample collection procedures. Lab personnel verify the specimen integrity, ensure proper labeling, and follow standard handling protocols. And support staff. They manage patient intake, facilitate specimen transport, and assist with electronic records. This is why it's a collaborative effort. All stages can be collaborative efforts, especially you most likely don't have one person going from end to end. It's very unlikely. So they're all pieces of this. And so there are multiple staff that can be involved in the pre-analytical phase, and there's multiple staff that can be involved in the analytical phase. So it's important to understand everyone's role. Everyone understands their role and to work together towards the same goal. Some key activities include test request and patient preparation, the accurate test selection and proper patient instructions, such as fasting, specimen collection, use of appropriate test techniques and materials. Examples include the correct collection tubes, sterile conditions, sample labeling and identification, to prevent misidentification with barcoding and double check the system. It's important to maintain the integrity of the sample. We have to know which sample we are testing and make sure that it's right with the patient. Typically samples come with a barcode on labels, tubes and things like that, and that can be carried on throughout the chain of custody, which has the patient information. The chain of custody should match the patient sample, the tube, and double checking that to make sure that it's correct, they match up. And specimen transport and handling. Maintain the integrity through proper temperature, timing, and handling protocols. Every sample has some kind of stability there's transport stability, and then there's overall collection time stability. Sometimes samples are sent through FedEx. They're mailed to locations. They have longer stability. Blood has very low stability. CBC sample has to be tested within 24 hours of collection. Typically, there's very few instances where that's not the case. And other analytes within blood or serum specifically, they do have a very short stability. So they're not stable. What happens is blood starts to break down. Even with the preservatives in a tube, things start to break down. Cells start to lyse, which creates changes in the sample. Either some drugs increase in concentration as time goes by, and some drugs decrease. I say drugs, I should say analytes, because they're not technically drugs. Your sodiums, your potassiums, those might increase as cells lyse, because they're lysing out of the cells, and it's an inaccurate estimation. HbA1c is one that does change too if the sample stays for too long, especially if it sits for too long. If it's not mixed properly, it's going to have an artificially high HbA1c. So sample handling is very, very important from a lab standpoint, and I think from physically what's happening in lab test results. The sample transport and handling is the most important, because those do impact results, and there's nothing that a laboratory can do to fix that. Once the sample's been past stability, once the sample's integrity, once it's no longer useful, once it can no longer get an accurate result, then there's no going back. There's nothing that can be done. The other issues, maybe if the form's missing some information, or they don't have a name, they don't have a signature, those can be corrected, and those pre-analytical spots can be corrected, but the testing, the stability of the sample, those cannot be rectified. If we go past that, then there's just no going back. Also, if the patient wasn't fasting, that's going to have a problem. It's going to have a poor result. But that's something that the laboratory doesn't really have control over. Won't even actually know that. And then, of course, if it's collected in the wrong tube, that can cause issues. Because a lot of, especially if it's like blood, then CBCs have to be in a certain tube to maintain the cells, because you're counting cells. If you're crashing it out, if it's in a serum tube, it's just not going to work. There's just no way. On the flip side, if you need to test serum and you collect it in a tube that has CBC, that is designed for CBCs, then that's not going to work out for serum. Because you need a serum separator, you need to test, you're validated and testing on the serum. You're specifically looking for the concentration of the analytes in serum, and so you need to have it in serum. So these are all things that are very important, and these are all aspects of the pre-analytical phase that really does impact the lab, and it impacts not just the laboratory results, but it also impacts the overall patient results. Some common errors in the pre-analytical phase include mislabeling, incorrect tube usage, poor patient preparation, or delayed transport. These errors and the errors that occur in the pre-analytical phase can be overcome, or they potentially could be unrecoverable. There's a saying, garbage in, garbage out. Our results are only as good as the sample that comes in. If we receive garbage, then our results are going to be garbage. So it's important that the sample we receive, it's processed in the pre-analytical phase, that it comes in the most pristine condition. This means we should reduce delayed transport. Anytime you have a delayed transport, you have potential for issues. Temperatures could be elevated. If it's left in a car courier transport, if it's delayed when it comes to being shipped, all these things can cause a temperature issue when it comes to stability, time issue. If you only have so much time before the sample can be tested to have accurate results, all those things. And then there are simple things, poor patient preparation. If the patient isn't fasting when they should be, the result's going to be accurate, but it's not going to be a useful result to the patient because they weren't fasting. The result might be accurate, but it's going to be a non-fasting result. And if we report out, if it's checked off as fasting, but they weren't fasting, they're going to get a result that doesn't make sense, and it's not going to be great. Mislabeling, that's the most simple one. If something's on the chain of custody, those can be corrected. But indirect tube usage, that's honestly usually going to be an issue. In the molecular side, it's not nearly as bad, but when it comes to blood, if they choose the wrong blood tube, the results are going to be toast. Errors in the pre-analytical phase can have multiple impacts. They can lead to inaccurate test results, unnecessary retests, and delays in diagnosis. Oftentimes, these errors in the pre-analytical phase are going to need to be retested. These type of testing in the wrong tube, storage delays in delivery. If they're outside the stability time, if they're outside the usefulness of the test, then there's no use for them. They have to be retested. That's going to delay the diagnosis. It's going to delay the time it takes for a patient to get results. Or worst case scenario, if it's hidden or if it's not tracked properly, it can lead to inaccurate test results. If there's stability issue, if you're testing a pathogen and it got to an elevated temperature for an extended period of time, and the DNA and the pathogens have degraded, you may not be able to identify it. On the flip side, if you have a CBC, your blood cells may have lysed, and you don't have an accurate count anymore. Also, cells lysing could cause serum values to be different. This specific certain analytes. Also, some analytes are more likely to bind to the lysed cells once they've sat for too long. So a lot of things in the pre-analytical phase can break it down. Like I said, garbage in, garbage out. You can only do the testing with the proper samples, and to get the proper samples, you need to follow everything within the pre-analytical phase. So, some best practices. It's important to request the test and have order accuracy. Ensure clear communication between clinicians and laboratory staff. It's important to order what you want to order. Oftentimes, a person or a nurse or doctor even, they may think they want one thing and they order another. So it's important to make sure that what the actual patient needs is being ordered. It's very easy to mix up analytes and blood testing, to test the wrong thing, and to request the wrong test. So it's very important to make sure that they're requesting what they actually want to see. So it's important, useful to use EMRs or electronic medical records to minimize the manual entry errors. Now, of course, you can still click the wrong button, but this does help. And specimen collection. Follow the standard operating procedures for vein puncture and other collection methods. This is important because it's important to use the appropriate collection chambers, so the appropriate tubes, and to adhere to patient preparation guidelines. It's important to make sure you're in the right matrix, so you have to use the right collection container. Especially if it's PCR or molecular, then you need to make sure you're collecting with the right collection device. So oftentimes the validation occurs with a certain collection device, so like a nose swab or urine cup or something like that. And if you have the wrong matrix, then technically the laboratory cannot test it, because you have to match your matrix to your validation. And then, of course, we discussed in length about blood tubes. They need to be the correct blood tubes, or else you might impact the stability of the analytes and therefore the overall results of the test. So it's very important to make sure that you're using the appropriate specimen collection. Also, label and identification. Implement a two-tier identifying system, patient name, patient ID. Use barcodes or RFID for automated tracking. This helps to eliminate errors in labeling. And then specimen transport. Establish strict temperature controls, ice packs for chilled specimens, and minimize the transit times with optimized routes or pneumatic tube systems. So if you're testing within a system, if your laboratory is within the clinic, then you can have tube systems, so you can just kind of like the old mail chutes, and you can kind of get the samples there. Or if you have a centralized lab and you have samples coming from multiple clinics, it's important to make sure that the samples are getting to the laboratory as fast as possible. In many instances, especially blood, you have to be there basically as fast as you can. And then, of course, to establish strict temperature controls. Often these tests are FDA controlled, and then they have specific stability windows to specific stability temperatures. So you can't be outside those temperature ranges, or you are no longer following the manufacturer's guidelines. If you're no longer following the manufacturer's guidelines, then you become a LDT, or lab developed test. Lab developed testing requires much more validation, much more intense validation. It's not just verification, as we spoke of in previous modules. You have to do a full validation. If you become LDT, if you're no longer following the manufacturer's recommendations, then you become LDT, and you have to do all the experiments, all the different types, stability controls, precision, accuracy, all that good stuff. You have to do all that stuff on your own if you're outside the manufacturer's guidelines. So it's important to know what the temperature ranges for stability are for your analytes. Some analytes are better at room temperature. Some analytes are better refrigerated. So depending on what your laboratory is testing, you may need to have transport in ice packs. Some laboratories receive samples by mail, and they can become at room temperature. But it's important to evaluate what happens if there's an elevated temperature. What happens if you have too high of a temperature? So those are all things that are important to you can minimize these issues and not have to go above and beyond if you can maintain control in your pre-analytical phase. You can always prepare, as a laboratory, you can always decide to prepare for the worst case scenario, but you can also try to limit these issues by having a shored up pre-analytical phase. And then, of course, communication and training. Conduct regular staff training on collection techniques and error preventions. Maintain clear communication between collection sites and the laboratory. It's important to have continuing education, and sometimes continuing education can just be retraining, revisiting issues, revisiting collection techniques, the containers that are used, all that good stuff. Refreshers. It's always important to have that, and it's also important to maintain communication between your collection sites and the labs. I think it can be very easy to have a breakdown in communication because they can be very separate. They can be very different, especially if you have a central laboratory with multiple clinics collecting. They can be very disconnected and not feel like one whole entity. So it's very important to try to maintain communication between the sites and the lab. If you're fortunate to be a laboratory inside a clinic where your teams can see each other much more, then you can have meetings. You can have bi-monthly meetings, monthly meetings, quarterly meetings, just something to establish communications. You can even have team building exercises, team building activities. So there's a lot of things, and it's important to... The final words for this session. Excellence is not a singular act, but a habit. You are what you repeatedly do. So always be learning, always be checking your processes, always be helping your staff improve, and teach your staff how to continuously learn, because you're not a singular act. Excellence is not just something that happens one time. It's a continuous process. It's a continuously what you do. It's not just one time. And that's the reason why COLA, why your CLIA, why it's so important for compliance, is because you must be able to repeat it. Repeat. Continuous improvement, that's the goal. Continually having, because if you're not continuously improving, then you're falling behind. And if you're falling behind, then you're no longer excellent. And with that, up next we have Module 10, Laboratory Management Simulation. Be sure to take your quiz and prepare for Module 10. We're almost done. You're almost finished with your Lab Director qualification. So, finish that up, and I'll see you in the next one.